And the main pathway, the pattern recognition receptor. And it kind of makes sense. If you block the pattern recognition receptor, you get more growth of the bacteria. So I don't know how I am in time. 2 minutes. So I just want to briefly summarize. This is very preliminary results. This technology has been used a lot in cancer research. More recently, Dr. Jonathan Kahn has been used very successfully this technology to find genes that are important to maintain HIV latency. And we think there's a future for this technology to study MTB infection. It has several advantages over the regular silencing RNA screen that I'd be happy to discuss with you, but we don't have time now. And the other thing that I want to point out, this is only 3 months of work, and we need to really confirm this data and do some statistical analysis, and also we had to do validations with single shRNA in both THP1 cells and macrophages. So very quickly switching gears, now on the other direction of the interaction between MTB and HIV. I just want to briefly summarize that, or the main finding of our CFR pilot award was that we found that the Mycobacterium PIM6, phosphoadenoacetate monocyte, which is one of the main components of the cell wall of MTB, that has also been detected in exosomes, which are microvesicles that are delivered by infected macrophages, upregulates TCR-triggered HIV replication. And this data was shown this year in the Keystone meeting, and we are close to publish the data. Hopefully in the next month or so the experiment, the final experiment will work, and we will be ready to publish. So what we found basically is PIM6, but none of the other cell wall associated glycolipids, LAM, LM, or PIM2, which is very closely associated with PIM6 in structure, induce increased HIV replication in presence of anti-CD3 triggering that was evident by P24 staining and also by doing the RT activity in the supernatants. This activity of the PIM6 was associated with increased interferon gamma, most likely with increased T-cell activation. And the mechanism, we think, is mediated through TLR2 because PIM6 was a very strong TLR2 ligand. And we are trying to confirm the mechanisms and do some intracellular signaling analysis to see what's the pathway that is actually activated. So, well, I'm going to skip that one. I just want to thank very much to the CFAR members at CASE, especially John Carn, because he has been a wonderful mentor. And he has really allowed me to set a different direction in my research. And I want to especially thank Ching Lee and Miriam Rodriguez, who are the persons that have done the experiments. And of course, I'm very thankful to the support funding from the CFAR Developmental and more recently the Supplemental Award.
Quick question or two. All right.
So the host molecules that you can knock down and get increased bacterial killing are really lovely potential targets for host-directed therapies. Mm-hmm. And you talked about thyroidine. I was wondering if there are other genes in there that look like there are existing drugs already targeting those.
Yeah, I look at every single sequence for that. And the answer is no, not from this experiment, this preliminary experiment. But first of all, we are using BCG. We are not using RV. So the goal is to use RV at some point because the pathogenic mechanisms are completely different. This is just to set up the system. And second, we do have another grant focusing on that, looking at host targets, using this technology combined with screening chemical libraries to look for host targets. Yeah.
Okay. Thank you. So next is Christoph Grundner from the Seattle Biomedical Research Institute, who's going to talk about phosphosignaling correlates in the MTB latency approach.
Thanks. It's nice to be here. Thanks for inviting me. I'm Christoph Grundner. I'm at the Seattle Biomedical Research Institute, and I'm in part funded by a New Investigator Award from the University of Washington, CFAR. My lab is interested in phosphosignaling, especially in how phosphosignaling regulates latency and reactivation. So this is a billboard I saw in Delhi earlier this year, and it reminds people to please take their full regimen of TB drugs unless they want to risk getting MDR TB. And this unfortunate person right here clearly doesn't have latent TB, but active TB. And that's really what we talk about most of the time when we talk about tuberculosis, active disease. But there is, of course, this. There are many more latently infected patients, and this provides sort of the reservoir that supplies people into these active cases. So what I'm really interested in is what happens at this transition. The molecular mechanisms are really poorly understood. And so we've been thinking of ways to find out really on a protein functional basis what happens when TB reactivates. So we actually chose an approach called activity-based protein profiling that I'll just introduce very briefly. ABPP uses active site-directed probes, and using these probes that covalently bind classes of enzymes based on a shared functional mechanism, you can then profile this whole protein by adding either, for example, a fluorescent tag through click chemistry or a biotin, and then do downstream mass spec analysis. So setting up this ABPP approach in the lab, we realized very quickly that we actually have a fantastic tool for functional annotation here. So what you can do with these probes is really high throughput functional protein annotation, and I think you can't really do that with anything else. So why do we care about annotation? So the TB genome was sequenced 14 years ago, and we still don't know what probably most of the TB proteins or genes do. So for example, 1/4 of all genes are hypothetical or code for hypothetical proteins. We don't even know if these are made. Of the genes that are actually annotated, 5 to 40% are annotated wrong. So that leaves us with about half of all TB proteins for which we have no idea what they're doing. And I think that's really a fundamental problem in understanding TB pathogenesis. So we decided to do functional annotation initially, and we chose this ATP-based probe because we're interested in phosphosignaling, and also because a lot of proteins are ATP binders, and we thought we can really cover the TB proteome very well with this probe. So it's basically an ATP with an acyl phosphate, and instead of dephosphorylation or phosphotransfer, when you add this to an ATPase, you add this acyl phosphate to the nucleophile from the protein. And with this click chemistry end, you can then attach a fluorescent dye or a biotin and further analyze. This is what it looks like when you add this ATP probe to TB lysate. You see a lot of proteins lighting up. You can compete this binding, not surprisingly, with ATP gamma S or ATP. DATP does not compete with the probe, so it is specific, and neither does GTP. So it seems to work very well. It's very specific. And so what are these proteins? And we used a quantitative mass spec approach to identify these proteins, the accurate mass and time tag approach. And this is sort of a heat map representation of the whole data set. This is one line is one protein with the replicates here. And we only chose those that were actually 5-fold enriched in the probe labeled versus control samples. So you can see the replicates look very similar. This is an, I think, important control, because with this ATP gamma S competition control, you can actually show that all of these proteins do bind ATP and not just some reactive surface lysine residue. So we identified over 360 ATPases, and what are they? Not surprisingly, a lot of them are known ATP binders, kinases, chaperones, other ATPases, acyl-CoA, and NAD/FAD binding proteins that we know recognize adenosine. And then there's this group of DNA RNA binding proteins, which puzzled us initially because we didn't quite understand if the probe binds as a cofactor or if it actually binds in the RNA-DNA binding site. So we know that DATP doesn't compete with the probe, so we parsed these a little further and saw that actually most of these are RNA binding proteins, for which it would make sense if the ATP probe actually binds in the RNA binding site. But the group of proteins we were, of course, most interested in are the hypotheticals, because here we can really do new annotation. There were 73 hypotheticals, and looking closer by bioinformatics, we could actually find for about half of them an indication of ATP binding just through looking at the sequence. But there was another group of 37, which we could really not find any evidence of ATP binding of any sort, and there was no really similar sequence out there. We expressed some of those to make sure we were really looking at ATP binders, and sure enough, they do light up with this probe. We can also identify the catalytic lysine residue. So although these proteins have no sequence that's in any way similar to known ATP binders, they use the same ATPase mechanism. And we were especially interested in a group among these hypotheticals of 4 essential proteins, because whatever they do, it's important. So we set out to crystallize those and got a structure of one of them. It crystallized as this dimer. And so does the structure tell us anything about protein function? Yes, it does, because surprisingly, the closest structural homologue in the PDB is actually in RNAs. And again, let me remind you that we could not find any sequence similarity by any way. So this was really surprising, but an RNA fold doesn't make an RNase. So we looked closer at the catalytic residues, which are especially these DEDD in this specific RNase family. And if you overlay the TB structure, you can see that all these residues are actually there in the TB enzyme, and they're also in the same place. So the electrostatic surface is very similar, also indicating RNA binding. And we then wanted to find out if ATP really binds as a cofactor or as in lieu of RNA. And so we got another structure with AMP bound, and sure enough, the AMP actually binds exactly where the RNA binds in the E. coli enzyme. So I think this was a really useful exercise. We really identified a lot of new ATP binders, and we validated annotations, in silico annotation for a lot of others. But what ABPP is really best at is at comparing biological samples. So going back to latency, we wanted to see what ATPases are involved in these transitions between replication and dormancy. And so we're very lucky in that we're next door to David Sherman. So we use David's hypoxia model, which recapitulates at least one of the major factors of latency. And we exposed bacterial cultures to low oxygen. They stop dividing, and then you can re-aerate, adding oxygenated air back in. So using this hypoxia system, we then did ABPP, and you would expect that after 7 days of hypoxia, when cells stopped replicating, that protein function would, or enzyme activity would actually go down. But surprisingly, what we saw is that we see a lot of protein functional changes in these ATPases that are like on and off events, and we're now doing the same quantitative mass spec analysis to find out what these actually are, and we're hoping to identify really players that mediate the switch, and also potentially novel drug targets that might make non-replicating bacteria more vulnerable. So to summarize, we identified over 370 new ATP binding proteins in TB. Many of those are actually new with completely unique sequences. We identified an RNase that is very similar to an E. coli RNase T ortholog, and we're now working on defining these ATPase functional changes we see in the hypoxia model. This is my lab right here. Most of the work I've shown is Corey's. She's a graduate student in the lab with help of technician Anya here. And David Sherman helped with the hypoxia model, and mass spec was done at the Pacific National Lab with Charles Ansong and Aaron Wright. And we worked with Jan Abendroth on the structure. Thanks.
Was this done with virulent TB or was that BCG?
There was H37RB.
This might be a very simple question, but it's not my area. But is it the use of a physiological stress like hypoxia to mimic latency? Is that well documented to represent the same sort of signaling downstream that you'd expect to lead to latency from immune from other reasons that you might use latency besides just physiological?
Yeah, I mean that's a long-standing discussion in the field. I think it is fairly clear that hypoxia is one of the major determinants for bacteriostasis in latency, but of course there are many others. And the molecular mechanisms and correlates are really not well understood, and that's one thing we want to really get at here.
So is this labeling approach limited just to extracellular fluid culture bacterial growth, or can it be adapted to intracellular bacterial labeling?
Yeah, so we've been thinking about doing it in infected cells, which would be fantastic to do. But you have very low amounts of TB inside and TB protein. So even mass spec is really at the limits there of detection. So unless you do selected reaction monitoring or some other really high sensitive mass spec approach, I think you'll have a hard time seeing differences. But it would be fantastic to do that, yeah.
Okay, good. Thank you.
Thank you.
So the final presentation in this session is from Frank Wolschendorf from UAB, who's going to talk about phagosome function mimetics in TB drug development.
Thank you very much. It's really nice to be here and talk about the research that we are doing in Birmingham, Alabama, within the CIFA Center on Mycobacterium tuberculosis. And I think I am covering today drug development on Mycobacterium tuberculosis. And we have identified recently a link between Mycobacterium tuberculosis, copper resistance, and virulence. And we identified a protein that we call MCTB, and the TB mutant for this protein didn't grow in the presence of elevated copper levels, and it accumulated more than 100-fold more copper than the parental strain Mycobacterium tuberculosis H37RV. And to our surprise, this mutant also had a very severe virulence defect in guinea pigs and also in mice. And these data indicate two things. One, Mycobacterium tuberculosis is exposed to elevated copper levels during the course of infection. And second, it requires resistance mechanisms to cope with these stresses. And what are the sources of copper in a human host? There are a few publications out there. One I mentioned here identified that Mycobacterium tuberculosis is exposed to elevated copper levels in the phagosome. Oh, that was the backward, sorry. And then a few years later, it was published that this increase in copper actually contributes to the bactericidal activity of macrophages. And to wrap this all up, we could show that the copper content of Mycobacterium tuberculosis infected lung tissue does also increase. And what are the resistance mechanisms? In Mycobacterium tuberculosis in 2007, the copper-responsive CSO operon was discovered, and this is controlled by the CSOR, which is a transcription regulator, and it controls an operon that consists of a putative copper exporter. And just like MCTB, this CTPV protein impacts virulence of MTB. And then last year, a publication came out where they discovered another copper-responsive regulator that is called RIG-R. It also binds copper, and when it does so, it releases it induces expression of an entire regulon. And some of these genes that it induces are also implemented in copper resistance, and others, they are important for virulence, but we don't really know what they actually are doing. So since copper is successfully employed by the immune system, or by the innate immune system, against bacterial pathogens, we were thinking of ways how to exploit copper intoxication of Mycobacterium tuberculosis for drug development. And the obvious way to go is to look for inhibitors that specifically inhibit copper resistance pathways or proteins of Mycobacterium tuberculosis to make Mycobacterium tuberculosis more susceptible to the copper-dependent innate immune functions. And then the other idea that we are actively pursuing is to identify compounds that deliver copper across the mycobacterial outer membrane, which is a very efficient permeability barrier, and it naturally provides MTB with resistance against a lot of toxic compounds, including copper. And because these compounds would mimic the phagosomal copper burst, we refer to those as phagosome function mimetics. And the strategy that we are pursuing is we developed a high throughput screening platform, and the basic idea of this screen is that we screen for compounds against Mycobacterium tuberculosis in the presence and in the absence of copper. The readout will be provided by Alama blue dye, and this dye is really convenient because it measures metabolic activity, and if these cells are viable and if they reproduce, then this dye turns into a pink compound it turns from a blue compound into a pink compound, and it also is fluorescent, and it emits at 590 nanometers, which can be measured quantitatively. And these are some of the predicted results. We're not really interested in those which do not have any effect on the growth of Mycobacterium tuberculosis, and these would reconstitute a result for general inhibitor, and we are adding common antibiotics to our assays as a negative control, and copper does not affect the activity of those. And then this is one of the hit scenarios that we are most interested in. It means that Mycobacterium tuberculosis grows in the absence of copper, but the growth is inhibited in the presence of copper. And we identified a group of compounds which are called thiosemicarbasones. And here's the structure. We had actually two of these compounds in our screen, in our pilot screen. They're called ATSM and GTSM. They are only different in these side groups around it. And what they have in common is they are copper complexing compounds. And they are currently investigated for other reasons, as a cancer agent or also for Alzheimer's disease. But what's most interesting about those is that these compounds accumulate in hypoxic tissue, and as we just learned, hypoxia is a feature of granulomas and MTB-infected lung tissue, but it can also be a feature of the microenvironment inside macrophages, MTB-infected macrophages. So very interesting compounds. And here's the activity profile of ATSM. As you can see, there was no significant activity against Mycobacterium tuberculosis in the absence of copper. But if we add copper to the assay, these compounds are quite potent. They act against Mycobacterium tuberculosis in the lower micromolar range. And Here's the profile for GTSM. It's a little bit different. It looks like as if GTSM can actually synergize with the trace amounts of copper that we have in our assay, and we need to provide those because copper is an essential micronutrient, so it facilitates growth of TB actually at those concentrations. And GTSM is way more potent. It is active against TB at nanomolar levels, which is about 250 nanomolar here. Another interesting feature about GTSM is that they are also working against non-replicating MTB cells, and we used a nutrient starvation model to generate those cells, and this is not really the best model for latent MTB, but it is a model that can be used for high throughput screening because we can generate these cells in large amounts. And we also tested toxicity of these compounds against a monocytic cell line. We used THP cells, and as you can see, I think this pen is giving up, they are quite well tolerated by macrophages. The preliminary therapeutic index that we calculated is greater than 16, which pretty much means that there's a 16-fold difference between the active concentration against Mycobacterium tuberculosis and the toxic concentrations against eukaryotic cells. To summarize my talk, we have identified copper homeostasis as a potential new drug target, and we developed and validated a high-throughput assay platform to identify compounds that interfere with copper homeostasis of Mycobacterium tuberculosis, and we identified ATSM and GTSM as first proof-of-concept compounds. So with that, I would also like to acknowledge everyone that was involved in this study, especially the Kutsch lab and the Niederweiss lab, and also our collaborators from the Kansas State University, Stefan Bosman, who synthesized these compounds for us in large amounts, and also Randall Basaraba from Colorado State, who did all the in vivo experiments that I was talking about today. And we have funding sources from the National Institute of Health and also from the CFA. Thank you very much.
Questions? Sarah?
So, do you have a sense of what the temporal nature of the copper burst is in macrophages? And are you, is the bacterial compartment always copper-rich, or is there just a brief window in time in which that copper burst occurs?
Well, that's a very good question. I don't really think I have a good answer for that. The copper burst that we see, that we actually think happens, we don't really know when it happens. So is it one of the early things that occur, or does it occur after Mycobacterium tuberculosis, for example, stalls phagosome maturation? We don't know at which stage this is happening. This is actually something we are investigating at this point. So, but it is definitely defense mechanisms of macrophages, and it kills generally a lot of bacteria that are not really pathogenic. So it's like Escherichia coli. Most of the studies were done with Escherichia coli, and they used copper-resistant mutants, and they